0. Hi friends, welcome to AGRI Classroom, the room where future graduates are trained. Today we are looking into the topic cell division, meiosis. Meiotic cell division was first described by Benvenuto in 1883. Observed in roundworm and ascaris. Let's start with definition of meiosis. Meiosis. 2 spindle-using divisions which reduces the chromosome number to half from diploid to haploid. Or the formation of 4 haploid daughter cells from a single diploid parent cell. Occurrence. In general, meiosis takes place during gamete formation in animals and spore formation in plants. In this flowchart we look in 2 different phases of meiosis cell division. Meiosis is divided into 2 main phases. Those are meiosis 1 and meiosis 2. In meiosis 1 is divided into 5 phases. Interphase 1, prophase 1, metaphors 1, anaphase 1, telophase 1. In prophase 1, there are 5 substages: leptotene, zygotene, pachytene, diplotene, diakinesis. In meiosis 2 is divided into 5 phases. Interphase 2, prophase 2, metaphase 2, anaphase 2, telophase 2. The detailed description of all the phases is as follows. Interphase 1 or pre-meiotic interphase. The interphase stage just prior to entry of a cell into meiosis is known as pre-meiotic interphase. During the S phase of pre-meiotic interphase, chromosome replication takes place. In this S phase 99.7% of DNA replication occurs. The remaining 0.3% of DNA replication occurs in zygotene stage of prophase 1. Most of the RNA and protein synthesis occurs in G1 and G2 phase of interphase 1. The remaining 1 to 2% of RNA and protein synthesis occurs in leptotene stage of prophase 1. First prophase. This stage is the longest in duration. This stage is divided into 5 substages. Those are explained below. Leptotene. Increase in the nuclear volume. There is chromosome condensation so that they become visible as fine threads. All the chromosomes in a nucleus appear wound around each other, like loose ball of wool. Proteins required for chromosome condensation are synthesized. Zygotene. This stage begins with the pairing of homologous chromosomes and ends when pairing complete. The remaining 0.3% of DNA replication occurs. In this stage, development of synaptonal complex. Pachytene. This stage begins when synapsis comes to an end and ends when homologous chromosomes begin to move away from each other. Further condensation of chromosomes and the chromosomes looks like shorter and thicker. Since homologous chromosomes have already paired, the number of observable chromosome is half the number of somatic chromosome number, which is haploid. Formation of chiasma in this stage. Crossing over occurs in this phase. Diplotene. This chiasma slowly moves towards end is known as chiasma terminalization. Chiasma terminalization occurs due to the movement of chromosomes away from each other. End of this stage, complete end of chiasma terminalization occur. Important point, don't confuse here. In pachytene, ends when homologous chromosomes begins to move away from each other, whereas in diplotene, movement of chromosomes away from each other is continued. Diakinesis. End of chiasma terminalization marks the beginning of the stage. Towards the end of diakinesis, the nucleolus and nuclear envelope disappears. Spindle apparatus become organized. Bivalents begin to migrate to the equatorial plate of cells. First metaphase. Bivalents are arranged at the equatorial plate. The centromeres of 2 homologs of each bivalent lie on either side of the equatorial plate. Spindle fibers from opposite poles are attached to the centromeres of bivalents. First anaphase. Separation of the 2 homologous chromosome of each bivalent marks the beginning of anaphase. 1 chromosome from each bivalent begin to migrate to the one pole, while the other moves to the opposite pole. The number of chromosomes at each of the two poles of a cell is exactly half of the somatic chromosome number. This stage comes to an end when the homologous chromosome reach the opposite poles. First telophase. The chromosomes uncoil only partially. Nuclear membrane and nucleolus reappear. In some species, example Trillium cell, enter directly into the second prophase from the first anaphase. Cytokinesis. In many species, example Meiosis, the cytoplasm of each cell divided into two halves by the end of first telophase. This two-celled structure is known as dyad. In some other species like Trillium, cytokinesis occurs only at the end of the second meiosis division. Interphase 2. There is no DNA synthesis during this interphase. In addition, chromosomes do not uncoil any further. Second meiotic division. During the second division, the two sister chromatids of each chromosome separate and migrated to the opposite poles. As a result, the number of chromosomes in each of the haploid nuclei remains the same. Therefore, the second division is referred to as equational division. Second prophase. The chromosomes are relatively much more condensed so that they appear shorter and thicker than in mitosis. There is further condensation of chromosomes so that they become thicker and shorter. The nucleolus and nuclear envelope disappears. Spindle operator is reorganized. Chromosomes migrated to the equatorial plate. Second metaphase. Centromere of all the chromosomes are arranged in a single plane. At the equatorial plate, the two sister chromatids of each chromosome are distinctly separated from each other, but they remained attached in the centromere. This stage is relatively quite short in duration. Second anaphase. Centromere of chromosomes divide longitudinally. The two sister chromatids of each chromosome begin to separate and move away to the opposite poles. This stage comes to an end when the sister chromatid of chromosome reach the opposite poles. Second telophase. The chromatids uncoils so that they assume the appearance of a loose ball of thread. The nuclear envelope is reorganized. Nucleolus reappears. Cytokinesis. At the end of telophase 2, the cytoplasm of each of two cells of a dyad divide into two parts. As a result, one parent cell produces 4 haploid daughter cells after the completion of meiotic division. As a result, the cell is appear like tetrad. Okay guys, hope you get everything. See you all in next video. To get further notifications, like, share and subscribe to AGRI Classroom.